Thank you. All right. So just to check up, Anna, do you see the right slide here?
It looks perfect, Kristian.
Perfect, thank you. So it's my pleasure to introduce to you then Dr. Mayr, Lorenz Mayr. He has 30 years of experience from companies such as Novartis, AstraZeneca, and General Electric's healthcare, but also venture capital experience from Syncona. Lorenz is currently the managing director for Biotech Consulting and the chief executive officer supporting the pharma and biotech industry. Lorenz's expertise covers everything from high throughput screening to new modalities like cell and gene therapy, with deep commitment to making innovative medicines affordable. So I look very much forward to get more insights from you where the science and industry is moving and how we at the universities can support the creation of new medicines. So I will hand over to you now, Lorenz, to share your slides.
Okay. I hope you can see this. It's in presentation mode. Okay, good. Good afternoon. Thank you, Kristian, for the kind introduction. I would like to I was asked by the team to describe my scientific journey, to present myself, and eventually to help people who are a bit younger in their career, to help them to progress their career with some of the learnings I've made over the last basically 30 years. And I was thinking about the title, and I took catalysis. I'm obviously a biochemist, so I like catalysis. And I was thinking about why catalysis is important for biochemistry and professional career development. In today's webinar, I would like to give a quick definition of catalysis, describe what I want to achieve. I will present a little bit about, very quickly, my own biography and work experience and where I have been and what my business journey has been. But the main pillar or the main part for today should be my 10 learnings. They are my very personal views. They might work and resonate with you or maybe not. And you can read them all afterwards when we share the slides. And of course, I would like to summarize this presentation and hopefully have about 10-15 minutes time at the end to discuss all together. So what is catalysis? It's basically lowering the activation energy of a chemical reaction. This is from the internet. You probably all know this. You have seen this in your biochemistry 101 class. It's basically you on the left-hand side, you have reactants. I try to point this out with a laser pointer. Oops, does that work? I'm trying to laser pointer. Yeah, you have a reactant, the starting material, the product which is formed. You have an activation, an activated complex, and a catalyzed reaction basically lowers the activation energy. And some of the key principles, I try to cut this very short, is you lower the activation energy by catalysis. The net change in energy is not affected, but you make the reaction rate of a catalyzed reaction much faster. And because the catalyst decreases the height of the energy barrier, it increases the reaction rate of both the forward and the reverse reaction to the same amount. So to remember, the catalysts allow a reaction to proceed via a pathway that has a lower activation energy. So it makes things, it doesn't change the equilibrium ultimately, but it makes reaction to go faster. And in biochemistry, we are all known by enzymes from the early 1900s about they can catalyze biochemical reactions significantly. And when you look it up in Wikipedia, we know there are about more than 1,000 catalysts or enzymes in the human body. And you should ask in your own career development, who are the people who can serve as a catalyst for your own career development, or where can you help when you're more mature in your career where can you help other people to catalyze their career development? And career development by catalysts could be the people, it could be locations, or it could be particular situations in your life. My own background is, you see it here, I was born and raised in Bavaria, southern parts of Germany. At my high school, I was at the German Military Academy, studied biochemistry, moved on to the US, studied molecular and cell biology, went back to Germany in biochemistry and biophysics, did a PhD again in Germany, then moved to the US, to Boston. And that was in terms of my academic training. In terms of my professional life, so when I came back from the United States, I started first in Germany at Bayer Central Research in Leverkusen. Then after 4 years, I moved over to Wuppertal Pharma Research, then further on to Novartis, as Kristian said, to AstraZeneca, in global roles to GE Healthcare, to Syncona. And I took a position as a professor at ETH in Zurich on genome editing. When I moved into a startup company, Vector, which unfortunately was liquidated just recently. And since about 2020, I'm running a consulting firm in Basel, and I'm working also since about 2 years in an investment group in Boston. So the important thing is that I've covered different areas like pharma, medtech, biotech, academia, venture capital, private equity. I can talk also about that and startup. And I've been working both at a beginner lab like myself, doing everything myself in the lab. I remember this quite well, up to very large organizations with several thousand scientists, which is a completely different challenge compared to working by yourself in the lab. So the company size, I was working by myself, and I was working, for example, in GE Healthcare as a senior executive in an organization which had a total of 450,000 people. At that time, certainly one of the largest ones, US company, one of the largest one. And I've been working, as I said, in Germany, in the US, in the UK, in Sweden, and in Switzerland, where I'm sitting now. I'm sitting in Basel. So which locations, where have I been? So the United States, I mentioned this before. I've been in Boulder, Colorado as a student, and in Boston as a postdoc. And across Europe, I've had the great pleasure to work in different countries, which are all in a way very different. And for me, every country was quite enjoyable. So obviously, I know Germany quite well, different parts of the country. I know Switzerland quite well, where I'm living now. The UK and Sweden have been extremely good combination, both in AstraZeneca as well as in GE. I spent most of the time going between the two countries, UK and Sweden, since large research facilities have been there. Now, a little bit about my background. So I grew up in a very small town west of Munich. It's shown on the map on the left. Very conservative area of Germany in Bavaria. You see some pictures, very old historic town, medieval town. When I moved over to University of Tübingen, studied biochemistry. It's a typical student town, has about 70,000 people and about 28 to 30,000 students, so quite nice. And I studied biochemistry there. Sorry. When I moved over to the US, as I said, to Boulder, that's probably my affinity for mountains and landscape. A very nice place, right at the edge of the Rocky Mountains, a bit north of Denver. Then I moved back to Germany, University of Bayreuth. Bayreuth is well known for that composer Richard Wagner, of course. Upper right-hand side, you see the opera house. But actually, the much nicer opera house is the old Baroque opera house from 1745. One of the nicest ones. We had a couple of castles, but also a very new university, which was formed 1975. And they basically, about 10 years later, we studied a major in biochemistry, and I took that opportunity to move to a brand new university, which was fresh, energizing, quite nice at that time. I moved to Boston I always had a passion for the United States. I moved to MIT, and I also got married in the US. I still have a very close and firm relationship to the United States. And that place has been particularly important for me since I met a lot of people which played a big role in my scientific life later on. I still have lots of contacts. What I didn't know at that time when I was in Boston, that the whole pharma world will change, and a lot of pharma companies, basically, historically, they have been in New Jersey and Delaware, and a lot of companies around the year 2000, 2002 onwards, with the move of Novartis, for example. Novartis moved out of New Jersey, like many other companies, and now Boston, of course, is the epicenter of the pharmaceutical and the biotech industry. And that continued in my personal life with Novartis, with AstraZeneca, with GE, and all the other activities. Then after my US journey, as I said, I moved back to Germany. Leverkusen, very industrial. It's well known for the big company Bayer in German, and of course football. If you like football, they were, a year and a half ago, or the previous season, we were the German football champion. Very, very industrial area, somewhere between Cologne and Düsseldorf. And it was a completely different world to the one which I was used to be in before. And then from reasons which I'll explain to you, which have been very important in my career development, I decided to go away from Leverkusen, from the headquarters, into the Pharma Research Center, about 14 kilometers away in Wuppertal. In Wuppertal, again, very industrial area. It has the Pharmaceutical Research Center. It has a very nice zoo. It has the world's first monorail, but also it has the world's first pharmaceutical drug. As you might see on the slide, it's aspirin. And aspirin was basically in 1897 by Felix Hoffmann a milestone, since he took a natural product and used a chemical reaction, acetylation, to make acetylsalicylic acid, aspirin in short. That was basically the beginning of synthetic organic chemistry for the pharmaceutical industry, so not too long ago. And from where I moved further down the Rhine to Novartis in Basel. In the meantime, there's that very nice campus, Frank Gehry building my office, for example, and labs was here. And Basel is a hotspot in the center of Europe for the pharmaceutical industry. And as you will hear later, that culture was very different to the one which I was used to be in before, and was also an important change in my life. Very much to my surprise, totally unexpected, when I moved over to the UK, and the AstraZeneca people did an extremely good job in pulling me over to the UK and completely changing not only large parts of my scientific thinking, but also my cultural understanding about the UK and Sweden. So I was based up in the northwest near Manchester at a place called Alderley Park. And the journey went on from a very nice area up there, a very nice research center, also to relocate down to Cambridge, a very academic place again, and of course Gothenburg in Sweden. And I really enjoyed it, as I said earlier, the constant change. Very often 1 week northwest of UK, 1 week in Sweden, 1 week in Cambridge, and sometimes also the US. It was an extremely rewarding time in AstraZeneca. And what I particularly liked were the cultural differences, and how well it panned out for me by working both with the Swedish and the British part of the organization. And I was very lucky to work at some very nice places, needless to say. And then after that journey, I got the opportunity to join as a senior executive to join GE Healthcare at a place called Amersham in the UK. So that's the UK place. You see Heathrow Airport. You see the place Amersham down here, a lovely area, very, very nice. And that's the hotel. If you remember the movie Four Weddings and the Funeral, it was actually the hotel where I stayed when I was working there. And the second half was GE Healthcare in Uppsala. And again, very nice university town. But also on the lower right-hand side, you see something which is very important for reasons which I'm happy to explain. They had over 80%, eventually up to 90% of the world's market for resins for biochemistry. And that is an amazing, if you ever have a chance, go to Uppsala, try to take a look, get a tour on site, which is now called Cytiva. This is absolutely amazing if you stand in front of that huge facility where 80 or 90% of the world's supply for chromatography resin is made in Uppsala. Yeah, okay. And then I took a position at the ETH in Zurich. I wanted to go back in academia. I had previously been for almost 2 decades a lecturer at the university in Germany. But I wanted to spend more time in academia. I wrote a grant proposal which was accepted at ETH Zurich, and I worked as a guest professor for genome editing. What you see here is the ETH in town up on the so-called Höngerberg campus. It's a bit elevated outside of the city. You see the city itself, and of course Lindt and Sprüngli, if you're interested in Swiss chocolate. And then I decided after that time that I also wanted to learn more about investment. Kristian mentioned I joined Syncona Investments right around the British Museum in London. I want to learn about venture capital, since I realized already quite a while back that the future in innovation will rely heavily on venture capital. And after that time, I moved back to Basel. You see the old historic part of Basel again, the new parts with the towers of Roche and the buildings of Novartis, the 2 big pharmaceutical companies in town, separated by about 2 kilometers, and the river. And of course, what's very famous, if you ever come to Basel in the summer, you can go swimming in the river and go with the current. Okay. Now I wanted to point out I was very lucky in my career to learn various technologies. You can go through the list, and very interesting fields. In 1995, I was in a project meeting where people basically challenged the value of monoclonal antibodies as a therapeutic. People actually didn't believe it at all. Unbelievable today. In 1998, I sequenced with a company from Germany the first eukaryotic genome worldwide. It's extremely small. It's only 6 megabytes. It was with the company Lion Therapeutics. I worked on HDS, ultra-high throughput screening in different formats. Back in 2004, I worked with a gentleman which I got to know in 2003 in Berlin, a gentleman from Yale called Craig Cruz. I started to work on targeted protein degradation with PROTACs. I worked on fragment-based drug discovery. Went on in AstraZeneca. I did various things, but some of the highlights where I learned a lot was phenotypic drug discovery in all flavors. Back in 2013, we had a very interesting collaboration between AstraZeneca and Moderna at that time to work on mRNA therapeutics. So mRNA not for vaccination, but as a substitute for protein-based therapeutics. Back in 2014, we carved out the AstraZeneca genome editing strategy. It was extremely interesting and rewarding. We worked on stem cells. At GE, I worked on PET tracers for imaging. I had this kind of a break in 2017. I moved from pharma biotech to a completely different field called medtech. And there was a reason for that transition. So I worked on PET tracers, cell and gene therapy. When I started a company back in Basel, was like particle-based gene delivery. Then had a bumpy part of my private life. So I had to look into immuno-oncology and precision cancer therapy. And then in 2014, I started working on longevity, healthy aging, And now I'm working in novel company creation and support organizations like Science for Life. And of course, I would like to continue for a couple of years and build new companies. So what are my learnings? Now, you have heard a lot about me, and I tried to put down 10 learnings, and I'm very happy if you want to challenge these comments. Learning number one, build a solid scientific base in your career. And what I had when I started, I had the pleasure to work with an extremely good PhD supervisor, and also here, the story here is I started in 1985. Hard to believe. So I started at the first university and had a curriculum which was 20 years old, from the year 1965. And I realized that can't be. I don't want to be a perfect chemist in inorganic and organic chemistry, but not enough on modern biochemistry. So I decided already after 4 weeks at the university to leave the University of Tübingen. It was a very bold decision. I said, I don't want to get to that old stuff. I want to have a very solid scientific base. And so I moved to the next university. And then after I got good training in biochemistry, I realized I thought quite some gaps in molecular and cell biology. So I moved to the US. And so my recommendation is try to get a very broad and solid scientific education. You need it, and it will be important for transferring knowledge from one area to another. So learning number one, build a very solid scientific base. It pays off in the long run. Yeah. Learning number two is follow the science. That was what I mentioned earlier. So I was a PhD student and had a supervisor who was solely driven by science, by rigor and scientific integrity, with zero interest in business and commercial aspects, no interest in politics, which is good and bad. But he basically told me, and he showed me, and I learned it, follow the data with utmost rigor and scrutiny, not the common belief of the textbooks. We had a paper accepted, and then we realized we had made a fundamental mistake. We had to pull back 2 publications, which were on the desk of a well-known Nobel laureate at that time, and we had to rewrite the scientific model. And what I learned is every scientific model will always be a good approximation to the truth, but never 100% correct. And your next model will be better. So what I learned as a PhD student, I think, was go and dig very deep in primary literature, discuss openly with experts, challenge the status quo, and try to follow just the data, not the scientific hype. And look for the unexpected and stay open-minded. And I think we eventually forget it in today's hyper-engineered scientific community. We really need to read scientific literature, primary literature, and dig very deep on every single problem. Next one, number 3, talk about the science. My postdoctoral supervisor was a superstar at MIT, extremely smart, extremely well connected, an excellent scientist, and a brilliant communicator, and just the latter was important. If you want to make a good career as an academic or somewhere in industry, biotech or whatever, you need to be able to communicate the science, talk about it, and you need to go out and talk to the scientific community. And when you work for a company which doesn't allow you to present your data, eventually you are not in the right company. It was one of the reasons why I left one company and, for example, joined AstraZeneca under the leadership of Mene Pangalas, where we had a very clear objective: go out, talk about the science, get exposed, get challenged, and publish in high impact journals. So, but again, go out and talk about the science. And the next one was a very bitter learning: avoid the losers. What I mean by that, I was working in one organization with, I think I can say, with a highly incompetent line manager. Yeah. and it was demotivating, time-consuming, and career-limiting. So after 4 weeks in that environment, I spoke to my +2 manager and said, "So what's going to happen now? Is one of us, my line manager, I have to leave?" And my boss, my +2 boss, obviously said, "Maybe you should leave." And I decided to leave the department. The department was closed when 3 years later down. It was not that I had predicted this. It was an extremely important learning. When you see that you're at the wrong spot with the wrong people, the wrong culture, the wrong science, have the courage to move on. Don't waste your time. Life is too short to waste your time with the wrong people. And always analyze the strength of your line manager and organizational unit. And by the way, also learn how to manage your line manager, but you need to find a good symbiosis with your line manager and your organization. And if there's, for whatever reason, not a good fit, then move on. Number 5, I call it go the extra mile. When you build a high performance organization, honestly, you have to work very hard. It's the quantity and the quality. I had the pleasure at MIT to get first-hand exposed to extremely smart people. Many of these people have made a brilliant career, mostly in the US, but also in Europe. And I was deeply impressed, also frustrated that everyone else was so much smarter than I was. But it was hard work and learning from people and their genius, I would like to say. And as one of my line managers later on in Novartis said, it's if you want to make a career in the industry, it's both the quality of what you do, but also the quantity. You can't do it in a 35 or 40 hours a week. You have to work hard if you want to make a career. So don't give up, stretch yourself and apply what we call in music or sports the 10,000 hour rule. You have to learn it extremely well. Number 6, make lateral career moves if needed. So I was once working within Novartis in an extremely well-engineered, well-established department. Everything was going very well. And I started to see minimum increase in learning. And eventually I started to get bored about my own work. Yeah, it was important not to show it that I got bored, but I had to find a new challenge. And when you should apply the 80-20 rule, but when you find out that you need to invest more and more and get less and less out of your job, you should ask, can you eventually deploy your resources elsewhere and to achieve more? And when I asked, sorry, at that time for a lateral career move, I had less people. I kept my salary. I had to start at a lower level in terms of organizational impact, but it was very important to go for a lateral career move and to learn something new. Yeah. And what I learned at that time, I call it the +1+2 rule. When people ask me, should I go the next step in my career? Then obviously you ask, okay, why would you do that? What can you learn? But you also need to ask the second next step question. So if you go with step 1, how does it change your position for the second next? Not that you rush immediately into that, or that you should start to think about it, but it should give you a better strategic position to progress your career and not going into a dead-end road. Okay. Now, number 7, I tried to indicate this earlier. When it's very important that you are willing, it was important for me, eventually for other people as well, to have a willingness to change jobs, employers, cultures, and countries. And when I was ready for a change and had opportunities, but not every opportunity was right for me. I had job offers where I realized somehow either the location or the culture of an organization was not the best possible fit. And so I didn't. And in other cases, I did. And I was, with every career change, I was extremely happy. And what I also learned back in the days in one of the trainings for the management, understanding cultural differences. Yeah, the different people in Europe, as well as the people in Asia, in the US, we have different cultures. And when you operate in a global organization, or when you try to solve also problems in a small biotech company, ideally you make very good use of the cultural diversity of the people. A Swedish person is different than a British person. A British person is very different to a German person. I'm not saying one person is better or worse. I'm just saying some problems eventually can be solved either by some type of people or some cultures better, or definitely in the team. If you bring the right people together, you get a synergistic effect. Yeah. So I was very happy to have a chance. And number 8 is to move to novel territory, leave your comfort zone. So I was having a very good career in AstraZeneca. Everything was going very well. And when a medtech company, GE Healthcare, came along and said, we would like to play in the big field of precision therapy. It was a very big concept. We would have merged our organization, GE Healthcare, which was the size of AstraZeneca at that time, with another very big organization, the diagnostics business, to become by far the #1 player on diagnostics and technical capabilities for precision therapy. So it was a very great opportunity. And I decided to jump ship, leave my comfort zone, which was pharma, biotech, drug discovery, and to move into medtech. And it was a very good learning. And later on, I realized, as I said earlier, I had a need to learn more about venture capital. Around, was it around probably 8 to 10 years ago, I realized that the innovation engine in big pharma was slowing down. They don't want to hear this, but I realized that more and more innovation came out of biotech, of startups. And that requires a completely different understanding, not only the science, but also how you manage and how you finance innovation in that context. So I had a chance to join an extremely good group, Syncona Investment in London, at a very nice spot, and learn it bottom up. I had no employees. I left a few thousand employees, and next day I was just by myself and had to learn the basics of the capital market. Yeah. But again, go to new areas, leave your comfort zone. And then number 9, second to last, pursue continuous learning. That's obvious. Everyone tells you that. But I found it over the years extremely rewarding to learn constantly something new. And I remember a day when I was with the colleagues in AstraZeneca in Gothenburg, and when someone asked me what my training was. So my training, as I said earlier, was protein biochemistry, hardcore biochemistry. And when I reviewed the department we had in AstraZeneca at that time, then I realized the majority of the technologies I was suddenly responsible for didn't exist when I got my PhD. There were no transgenic animals. Yeah, there were no recombinant proteins. There were no iPSC, and. And the only way to get job fulfillment, excitement, rewarding time is by constantly learn something new. And you have to implement this, everyone tells you, and take the challenge of learning as a huge opportunity. And when I jumped ship from AstraZeneca to GE, I think I was 52, and my son hugged me and made that very harsh and direct comment, which I've spelled out here at the bottom, and said, "Dad, I'm very proud of you that a man of your age still has the courage to learn something new." I felt a little bit intimidated or offended by that since he was referring to my age, obviously. But after a while, I started to appreciate that comment, since actually he didn't mean it in a bad way. He meant it in a positive way. He said, "Hey, I love you, I like you, but you are still willing to learn something new." And now, and the last one is you need to become a great teacher. So I realized it was back in probably the mid to late 1990s that East Germany was building up, and they need to have support. And I got the opportunity to become a lecturer at the University of Halle-Wittenberg, a very old German university, one of the oldest ones, not very well known for people in the West. Halle has about 350,000 people, about 25,000 students, and we had a very good biochemistry department. And when I started to expose myself, which is very different to become a university lecturer, and later on, of course, at ETH, the same, when you become a teacher at the university, you need to rethink your business and ask, how do I teach the people? How do I convey that message? And when you are a line manager in industry, you need to evolve. You start by yourself, or you have one or two employees, but over time, you need to educate your people to avoid that standing still phenomenon. And if you're a great teacher, ideally you should also lower the activation barrier for others to pursue their career. And short term, you might say, oh, why should I make this investment? But long term, it pays off with a very high dividend. So that brings me back to the take-home message. You should try to lower the activation energy for yourself or for others. You should actually catalyze reaction. You should make things which will, with no doubt, happen. You should make them faster. And that is the concept. And at the end, I would like, so that we have enough time, I would like to give some acknowledgements. A big thank you to my PhD and postdoc supervisor. They deserve a lot of credit. And the many people where I had the pleasure to work with them. I learned so much. I don't want to list anyone particularly. And again, that's all for now. I would like to stop here. You see again on a single page the companies and organizations I was working on, but you can also see my work areas. And I would like to stop here and take questions from the audience. Thank you so much. And Kristian, I stop sharing.
Yes, I think Kristian is muted, but Kristian will stop recording.